Hello students, welcome to Biotonic NEET channel. So I am Ms. Kopika, your biology teacher, your master teacher. So students, we are going to do molecular basis of inheritance. So let me know if I am visible. Yes, I am visible, right? Yes, okay, great. Yes, I'm visible. Okay, let's start the class. So last class, students, we were trying to understand the basics, right? The basics of DNA. What is nucleotide? What is, polynucleotide? And what is, so many things we understood. Correct? Today's class, we will be understanding about the experiments that actually evolved or helped in the finding of DNA as a genetic material, which were the properties, why we call DNA as a genetic material, and we will also understand in detail the central dogma, then replication, so all of that. Okay, so today's class is very, very important. So those who are new to this channel, quickly do subscribe to the channel, and if you want this notes, please do. You have to join Vedantu NEET English, or you can join Biology by Gopika. In both the places, you will be able to get the notes. Is this clear? Okay, fine. Let's start the class, students. So yes, those who are preparing for 12th, right, NEET, also that is your CBSE exams, also this will be really, really helpful. Okay, yes, fine. Okay, let's start the class. Quickly smash the like button, all of you, and mark your attendance. Okay. Hello students, hello. So when we talk about the experimental setup, the first ever experiment that came into existence was your Griffith's experiment. Okay. So Griffith's experiment, let's see what was Griffith trying to do. Okay. This mic is disturbing me. Okay. Students, my voice is audible, right? There's no problem, I believe. Yes, mic it's clearly audible. Okay. Yes. So students, what was actually happening? So Griffith's experiment was based on Streptococcus pneumoniae. So he was working on Streptococcus pneumoniae. Okay, he was working on Streptococcus pneumoniae, and he noticed that this Streptococcus pneumoniae has two strains. Okay, two strains. So all of this will, students, when we do different types of experiment, when you're working in your, when you're doing your masters or when you're doing your degree, you will always work on different types of strains. Okay, not everyone will take the same strain. Now, so imagine I am working on one strain and Banot is working on one strain. Both of us will have different strains, and the results also will be different. Okay, so always remember that there will be different types of strains. Okay. So Streptococcus pneumoniae, he noticed that there was a rough strain which was called as R strain. Okay, rough strain. And he noticed there was S, or your smooth strain, from the name itself. Okay. Now, why did this difference happen? Why there was a rough strain and why there was a smooth strain? This was because, okay, this was because of the presence of your mucopolysaccharide layer. Okay, it was basically a mucus sugar layer. Okay, a sugar layer which was present, which made the difference. Okay, this is okay. Yes, which made the difference between the R strain and the S strain. So R strain did not have your mucopolysaccharide, or you can, if you cannot remember muco, at least write polysaccharide. Okay, at least remember polysaccharide. And what happens, students? This rough strain did not have this mucopolysaccharide, and because the strain was not present, maybe because the reason is still not known, but then they say that because of the absence of this mucus polysaccharide layer, the rough strain was not virulent. Now, virulent means something that has the ability to cause a disease, okay, that can cause an infection. So smooth strain had this mucopolysaccharide and the R strain did not have it. Okay, so the R strain would look somewhat like this and the smooth strain would look somewhat like this because of the presence of this mucus layer. Okay, so he was testing on this mice. Mice is the only person whom we can test it on. So when he was testing, first he tested the rough strain and it is non-virulent. So when he tested it on the mice was living because it is not going to cause any infection. Okay, so the mice was happy, no problem happened. Okay, then the smooth strain. Smooth strain was added, and we know that the smooth strain is virulent, right? Because the smooth strain is virulent, we expected that the mice would die. So when we injected this to the mice died. Okay, both the results were as expected. So Griffith was not at all, astonished or anything. He was like, okay, chalo, this is what I expected. So this is what has happened. This was the thing. Now, Next, he was like, let me do one thing. Let me do one thing. Because this smooth strain, maybe because of this mucopolysaccharide layer, or because of anything, that component that is present inside them, this S strain is actually very active. Let me do one thing. Let me heat kill this strain. Okay, because students, you know, heat can kill almost everything. That is the reason why we boil water, why we boil milk, because we know that if there are any germs which are present in this will definitely die with the help of heat, right? So he was like, chalo, I will do one thing, I will heat kill this. Okay, I will heat kill this. And he heat killed the smooth strain and injected into the mice. Mice lived. This was expected. He's like, okay, with the heat something died or something happened. Okay, and the mice lived. Now he was like, okay, let me do one thing. There are two things that did not affect the mice. He did not want to kill the power mice. Okay, he was like, so sad, let me not kill the mice. He was like, okay, let me do one thing. Let me use the two things that did not actually kill the mice. Okay, which was the two things that he did not kill the mice? Come on, all of you, let me know in the chat box. Which were the two things that did not kill the mice? The rough strain and the heat kill smooth strain. Yes or no? Agreed, all of you? If you agree, smash the like button. Come on. All of you are capable of marking your attendance. Is it? Okay. One second. Yes. Yes. Okay. Good job. So students, we all understood this much? Yeah. Okay, so what was happening? They used heat kill, right? Yes. So they used rough strain with heat kill smooth strain. And what was expected? These two people were not causing any harm. Right? They were not causing any harm in the mice individually. When this both were injected individually, both were not causing any harm. Yes or no? But suddenly what has happened? When we injected this, suddenly the mice died. And then they were like, Griffith was so confused, because Griffith was expecting that heat kill smooth strain did not cause any harm to the mice. Rough strain also by individually did not cause any harm to the mice. Now he was confused, like, what happened? What really happened? I used the same thing. I used two formulas that did not kill the mice. How did the mice die? Then he was very confused, very confused. He did not know what to tell, what to do, right? You know, that situation. So when he used S, okay, let's write heat killed S strain, right? Okay. When he used heat killed S strain, okay, heat killed S strain plus rough strain, these two people individually did not cause anything to the mice, so we expected that the mice would live. Okay, but the mice died. Now he was confused, and after some confusion, he's like, okay, I think I understand what happened. He understood that from the heat killed S strain, right? This is the S strain. Okay, this is the S strain. He understood from the heat killed S strain, something got transferred or transformed into the rough strain, okay, R strain, making this R strain virulent. Making this R strain virulent. It is like if some person is always telling negatives. Always negative. What happens? You stay with that person for a longer time, you also become negative. Yes or no? Same thing happens, students. This S strain, okay? S strain, whatever was there in the S strain, right? That got transformed or changed to the R strain, and that R strain became suddenly virulent. Okay, so you should always stay with positive people. Okay, always surround yourself with positive people. Wherever you see unnecessary negativity, always move on from there, because you know it is people with so heavy heart that they spread negativity. Okay, normally people cannot do that, but people with very, very negative thought process, lifestyle, so those people spread negativity. So always be, even when during your NEET preparation, it's going to be a very difficult journey, but always fill yourself or be with be with people who are positive, who will spread positivity, telling them, no, it's okay, you can do it, Banod, or no, it's okay, you can do it, Vijayan. All these people, who tell positively, yes, you can do it, dear, no matter what. You are in this journey and you're not alone in this journey. Always surround yourself with that kind of people. Okay? Done? Yes. Okay. Great. Now, students, what is the thing? So this he understood, but he was not able to label, or he was not able to put a name for all this, right? He was not able to put a name for all this. So he told that from the S strain, something got transformed into the R strain, and that thing he called it as transforming principle. Okay, he called it as transforming principle. So, because Griffith did not know what exactly transformed and what exactly made the R strain virulent, he just simply called it transforming principle. Fair enough, right? So, yes, some people tell the virulence is because of the S strain's virulence is because of the presence of the mucus layer, okay, the mucopolysaccharide layer, but it does not prove it right? Because we know that something that transformed could be DNA, and DNA is not something that would get heat killed. Okay, so this kept continuing, students. Many other scientists came behind them, and many scientists was like, okay, let's do one thing, let's actually find a solution for this, right? Let's actually find a solution for this, or let's understand what was this transforming principle. So then the discovery was after the transforming principle. Okay, the discovery was after the transforming principle. Okay, clear or not? Yes. Okay. Now, Oswald, Avery, MacLeod. Okay, this is so Avery, MacLeod, and McCarthy experiment. This is how the experiment is known. Okay, so it is Avery, MacLeod, McCarthy experiment. Here, what was happening, students? They were working on the biochemical nature of who? Of the transforming principle. Very, very important question. They can ask you this. Many PYQs have been asked from this. Why? Because in your NCRT the experiment is given, but it is not very, the clarity is not there, the clarity is not there at all. Okay, it's just given in a paragraph, and that paragraph does not justify. Okay, the paragraph does not justify. No, Vijayan, I'm not at all. Okay, yes. Okay, so clear. So biochemical nature of transforming principle. Now, students, what is happening? Let's try to understand. So imagine there is, so now what are we working on? Biochemical nature of transforming principle. What is the combination that we are taking? We are taking the heat killed S strain. We are taking heat killed S strain plus R strain together, right? We're taking this combination. Why we are taking this combination? Because we need to actually find what has happened, like who went from S strain to R strain, right? So we are taking this combination first. So first, they basically, students, how did they do this? They basically took some kind of enzymes. Okay, they took 3 or more enzymes, but only 3 is mentioned in your textbooks. So they took 3 enzymes. Okay, yes, so they took 3 enzymes, students, and those 3 enzymes, they worked on those 3 enzymes. Okay, so what did they do? First, they took heat killed S strain and R strain, that combination they took, and they first reacted it with protease. So protease is an enzyme that can degrade protein. Simple as that. It will degrade protein. Yes or no? Let's also write it here. So this was expected to degrade protein. Now, when it degrades protein, imagine in this combination, heat kill S strain and R strain, imagine in this combination, protein was the one who got transformed. Then the moment we remove the protein, that means with the help of protease, we remove it. Problem sorted, right? Problem sorted. We understood who is the transforming principle. So they did the experiment. What happened? Protein got degraded. Still, they injected this combination. They injected this combination into the mice, and the mice died. The mice died. How did the mice die? If protein was the genetic material, then the mice should have lived. But here, twist happened, right? Twist happened. Now again, they took the same combination, that is your heat killed Okay, heat killed S strain plus R strain. Okay, and again let's write it again. Heat killed S strain plus R strain, right? Okay, so students, next they used RNAs. What did they use? RNAs. Okay, let's write it with a different color. Okay, they used RNAs. Now when they used RNAs, what is expected? They would expect the same thing. If RNAs, this will, what will it do? It will degrade RNA. So if RNAs was the genetic material, then what is expected? Simple as that. The mice, after, if we inject this, okay, if we inject this to the mice, here RNA has almost vanished. Okay, here also we can write protein is not present. Right, protein got degraded, so protein is not present. Here RNA is absent. Okay, so we inject this combination into the mice and the mice still dies. Then they were like, okay, fine, something is wrong. Then they told, okay, let's add DNAs. What is DNA is going to do? Degrade DNA. So here they added this into the mice. So here what is happening, students? The genetic material, the DNA is absent. So that time they injected into the mice. Okay, mice they injected. The mice lived. Or the mice is alive. Okay, let's use the word alive. The mice is alive. Now they were like, okay. So in this combination, right? One second, yes. Okay, heat kill S strain and R strain. In this combination, they were like, okay, now I understand. Something has happened, right? What has happened? So what they expected. Is this clear? Is it not? Is it stuck? No, right? Okay, students, is this clear? Let me know. So students, what is happening? First they took heat killed S strain and R strain. This was the combination used by Griffith. Okay, yes or no? They are trying to find the transforming principle. Clear. They first added protease. If protein was the transforming principle, then definitely if protease is there, it will degrade the protein, and if we inject the same combination without protein into the mice will die. Okay, the mice were supposed to live, because why? If protein was the genetic material. Then they did more experiments. Okay, again heat killed S strain and R strain they took, and they used RNAs, because protease, okay, now nothing can be done, the mice died. Okay, again they took RNAs which will degrade the RNA, and they injected that combination into the mice again died. Then they were like, let's do one thing, let's use DNAs. Okay, when we use DNAs, the DNA was absent and the mice lived. Okay, the mice lived. Okay, clear? The mice lived. So here they understood that yes, DNA was the genetic material. Okay, DNA is the genetic material. Yes, this is the conclusion. Why? Because DNA was absent. Mice lived. That means something that transformed from the S strain to R strain was DNA. So the thing that transformed from your S strain, right? Yes, from your S strain to your R strain, what was transformed? DNA was transformed. DNA transformed. But that was not the problem. The problem was that many scientists was not okay with this. They were like, wait, how are you saying this? What is this experiment? It does not make sense. So there is some problems. Okay, so then they were not ready to agree this, because they were like, maybe there is some error or maybe there is something. They were not ready to agree. That is when Hershey and Chase experiment came into existence. Okay, Hershey and Chase experiment came into existence. Okay, so students, look here. This is the diagram given in your NCRT. So first we will try to understand what all things did they take. So first, the students, they took bacteriophage or bacteriophage, however you want to say, you can tell. Okay, bacteriophage or your bacteriophage. What did they do? They are actually virus that infects bacteria. They are virus that infects bacteria. Yes or no? Agreed? Virus that infects bacteria. And their structure is such a way that they have a protein coat. Two things they have mainly. They have a protein coat, which you would be studying in your biological classification. Okay, you will be studying about capsomeres, capsid and all that, right? So students, those things, okay? Yes. Okay, those things clear? Students, if there is any spam, please just off the chats and just move on with the class. Do not bother. Okay, there'll be some people like this. I told you, don't keep yourself around negative people. Okay, just block the chats and just block that person and just be in the class. Okay, do not bother. Fine. Okay, so students, is this clear? So what is happening? This bacteriophage, that is the virus infect bacteria, right? The virus that infects the bacteria has two parts. That is your protein coat and your genetic material. Protein coat and your genetic material. Yes or no? Now this protein coat and genetic material are going to be radioactively labeled. That means we are going to radioactively label this. Radioactive labeling we are going to do. Both of them, okay? Both of them we are going to radioactively label. How are they radioactively going to label? See, look here. Radioactively labeled by S35. Okay, radioactively labeled by S35. And then radioactively, here it is labeled by phosphorus 32. Now students, what is happening is, why did we take specially S35 and why did we specially take P32 is because this P32 will be only present in DNA, because phosphate is only present in DNA. Okay, and sulfur is only present in protein. This was the confusion, if it's protein or DNA. RNA, they never had a confusion. Main confusion was, is it, yes, very good, Sandeep. The confusion was that, is it protein or DNA? Because they already knew about RNA. RNA is the initial genetic material, right? It is the one that gave the, help for DNA to be the genetic material. So it is always considered as the first genetic material, okay? Yes. So S35 is present only in proteins, okay, and P32 is only present in DNA. Now we have a clarity. What we should do further. Now they radioactively labeled it. Students, radioactively labeling means what is happening? Okay, radioactively labeling means what is happening? Yes. Okay, is this clear? One second. Yes. Okay. So students, radioactively labeling means what? We are growing them in a culture medium which has sulfur and phosphorus, okay, which is having sulfur and phosphorus. Now when they radioactively label means what? They take up that, right? We cannot like write it on them, no? So they will get radioactively labeled. So we can see during centrifugation, which is the heavier particle, which is the lighter particle, that is called as gradient centrifugation. That means depending on the densities at which they are present, we will be able to see the difference. Clear or not? Okay, clear. Fine. Now students, look here. So we understood this much, protein and DNA. Now they have got radioactively labeled. You can see radioactively labeled protein is red in color and DNA is here shown red in color. Okay. Now both of them are bacteriophages. That means virus that infect bacteria. And which is the bacteria we have taken? We have taken E. coli. We have taken E. coli. Now they were allowed to infect on the E. coli. What will they do? Virus nature is that as soon as they infect, they will leave the genetic material inside. That is the first thing about virus, right? So here you can see first step of your Hershey and Chase experiment is infection. First thing is infection. Clear? Now, they got infected and you can see immediately they are dropping their genetic material inside. You can see genetic material has gotten in both the side and it is very, very visible that the radioactively protein is already outside, right? So here students blending or using a mixer okay, mixer blender is to remove, mainly this step is done to remove the protein coat. To remove the protein coat which will be attached to the E. coli cell. Okay, to remove the protein coat. Okay, which will be attached to the cell wall. Okay, clear? Now, during the blending process or during the mixing process, the protein coat just vanished. Okay, tata bye bye, we told, it went off. Okay, now students, what happens? Here you can see, I told you, radioactive labeling is to check the centrifugation gradients, right? So we centrifuged it and we are able to notice, okay? So during centrifugation, always remember two things come during centrifugation. Okay, this is a tube. Okay, this is a tube during centrifugation. In this tube, and let's draw here also. Here is visible, huh? No, I have to draw it on the top because my head will cover you people. Yes, this is one tube. Okay, this is two tubes, students. This is one tube number one. That means where protein was there. What did they notice? They noticed that, wait, let's put black color. They noticed that there was supernatant. Supernatant is the portion which is on the top. Okay, the liquid portion, and the heavier particle is the pellet. Okay, so imagine pellet was there. Okay, now they noticed that in the supernatant they saw the protein, or they saw S35. That means the protein was present in the supernatant and not in the pellet. What does this show? That means protein is not the genetic material, right? If the genetic material is there, students, if protein was the genetic material, it would always be heavier and it would always be seen in the pellet. But then they understood that no radioactivity detected in the cell because the protein coat has the radioactivity. That went off in the mixer, right? It did not get transformed. Nothing got transformed inside the cell. That infection did not happen because of the protein coat, right? So no radioactive detected in the cell, and radioactive was detected in the supernatant and not in the pellet. This comes to a conclusion that protein is not the genetic material. Not the genetic material. Okay, clear? Protein is not the genetic material. Okay, clear? Yes, Vijayan, please block the spammers, and please, I have just focus on the class. Please off the chats. I'm not even looking at the chat, so please, yes. Okay, don't get disturbed with all this. Such kind of people will always come. Okay. Yes. One second. Yes, okay. Okay, done. So students, what is happening? Centrifugation, right? Centrifugation. So what is happening? When we added into the mixer, the protein coat got removed. Okay, now look here. Centrifugation, if you see, basically I told you, no, centrifugation only we will be able to understand which is the supernatant, which is the pellet. That's all. Simple as that. Now, centrifugation is basically done to understand the gradient difference or the density difference. Okay, so here what is happening? Look here. Look here. Okay, let's just draw it with the same color, red itself. So no, let's draw it with black. Okay. Yes, and here we have the pellet. Here we have the pellet. Okay, students, what did they notice? They noticed that the genetic material was in the pellet. That means your DNA was in the pellet. That shows that DNA is the genetic material. Yes or no? Shows that DNA is the genetic material. Simple as that. Okay, simple as that. Clear? Okay, so DNA was in the pellet, and because of this, DNA was the genetic material. Simple, that's all. This Hershey's and Chase experiment was actually proven and really accepted widely by everyone. Okay, because always understand genetic material is heavier and genetic material will always settle in the pellet and it will not be seen in the supernatant. By this, they understood that protein is present in the supernatant and it cannot be the genetic material, but DNA was present in the pellet and DNA was the genetic material. Okay. Yes, here you can see the same thing, students. I've just put a better picture. So no radioactivity enters the cell. Can you see? No radioactivity enters the cell because this was the radioactive substance and they are going out of the cell. Okay. Phage grown with radioactive sulfur, then centrifuge, radioactivity in the supernatant. Here same thing is happening. Radioactivity enters the cell. This will show that something is getting transformed, right? Something is getting transformed. Simple as that. Okay. Done. And phage grown with radioactive phosphorus. Okay, phage grown with radioactive phosphorus, centrifuge and radioactivity in the pellet. DNA is the genetic material, hence proved. Is this clear? Now we are going to the next topic. Are you all ready? Are you all ready? Yes. Ready, students? Can you tell me? Yes, yes. Okay, fine. Okay, so one second. Let me. Yeah. Okay. Yeah, at least know the gender. Yeah, Sanvi. It seems, he is not eligible to take the class. You're talking about yourself? Yes, you're definitely not eligible. Okay. Yes. Okay, students, do not bother, just block and move on. These are all useless people. And this is a YouTube medium, lot of distractions will be there. Okay, just block and move on, students, please. Do not get it bothered. Yes, okay. Now next we are going to understand properties of genetic material. Okay, a molecule. So now students, like for everything we have criteria, yes or no? To be a teacher we have criteria, to be a good student we have criteria, to be a spammer you don't have any criteria. So all this is there, right? So properties of genetic material, to be in, to do election you have criteria, to be like to be MLA, MP, all this you have criteria and you have some things that you have to achieve, some things that you have to do, right? To be a teacher, you have to make sure that the student understands, right? This is all about a teacher. Same way, to be a good student, you have to respect and you have to, be obedient. Same way, to be a good MLA or MP, you have to have certain, you should be an Indian citizen and all that. Same way, to be the properties, to be genetic material also, there is some criteria. Yes or no? Okay, to be a genetic material also there is some criteria. What are the criteria? Let's see. It should be able to generate its replica. That means it should be able to form its own replicas. Okay, like duplicates or copies, right? It should be able to form its own copies, right? How do they do it? DNA will be doing it with the help of semi-conservative method. Semi-conservative method. Then it should be stable chemically and structurally. Very, very important thing. And I told you yesterday's class, if you remember, I told you about this thing that it should be chemically and structurally stable means what, students? It means that that RNA, RNA has the presence of an extra oxygen, right? Presence of one extra oxygen. Because of which RNA is not stable. Okay, RNA is not stable, but DNA is more stable. And structurally also, the presence of thymine, okay, instead of uracil in DNA also makes it very, very stable. Is this clear? Okay, it should be stable chemically and structurally. That means RNA has the presence of an oxygen, which makes it actually really unstable. It makes it really unstable. Okay, and presence of thymine in DNA causes its stability. Okay, uracil is there in RNA, so that also makes the differences. Okay, now it should provide a scope for slow changes. Now, what is happening? That means slow mutation. Students, why do we need genetic material? That means over ages, right? Like now we are all growing old, right? Every year we are adding one age. Now, all those times also slow changes should happen. Not like, oh, today you're like, 10, tomorrow you're 20, suddenly all changes will happen. No, that will not be okay for our body. So, slow changes should happen. Slow changes with age, with, situations, with environment. All this should be very, very slow. So mutation is required, then only evolution will happen, correct? It should be able to express itself in the form of Mendelian characters. So Mendelian, according to your principles of inheritance chapter, you will know about your Mendelian characters. No, those things also should be very, very important. That means they should be able to inherit. They should be able to inherit. They should be able to do certain changes depending on generation after generation. Okay. Now we will see your NCRT line what is told. So students, very, very important words we will underline. Okay. The genetic material should be stable enough not to change with different stages of life. Right, we have juvenile phase, we have adult phase, we have teenagers, right? We have different stages, we have different stages in our life. All these times also our genetic material should not change drastically, it should change slowly. And mean, see, when you're in your teenage, right, you have this hormones released, you have this, you will like you have so many changes, like you will be sexually trying to look very beautiful, like you will be trying to become handsome or beautiful, you will try to look attractive to the person you like. All these changes, all these hormonal changes, when does it happen? In your teenage, right? That time if your DNA is changing drastically, then that means you will become like a different person, right? All the things in your body also will become different. That should not happen. Slowly it should change, that okay, this person has increased 1 year in age, some kind of changes have to do in the body, that's all. Okay, now different stages of life depending on the age or with change in the physiology of the organism. Okay, depending on the physiology of the organism. If you notice at one age, you become really mature, right? You will be in an age, if you're all like future doctors, if you're all preparing for NEET and you're future doctors, you will be in a situation that you will focus on yourself and you will be like, no, I do not care what is happening outside, I will focus on myself and I will take care of myself, right? Maturity is when you realize that these things, half of the things that happen does not even matter. Okay, so people like this spammers, no, those are not mature, they're like kiddish people. Okay, so that means what, students, change with depending on the physiology should also happen. Okay, now stability as one of the properties of genetic material was very evident in Griffith's transforming principle. Okay, so you notice that stability, that means no matter how much did Griffith heat killed, DNA did not get affected at all. DNA did not get affected at all. Even though he heat killed the S strain and kept it with the R strain, the heat kill S strain still got transformed, right? Still there was transformation. Still there was transformation that occurred. So stability is very, very important. Stability is very, very important, and it was noticed during transforming principle itself that which killed that heat which killed the bacteria itself did not destroy some properties. Students, not all the properties, some properties. That is the reason at first stage when the heat killed was injected into the mice died, right? Because some property was there to kill the mice. But at the same time, when we combine that S strain with the R strain, some property got transformed that made the R strain itself virulent. Okay, some of the properties of the genetic material, two strands being of genetic material, this is now can be easily explained in the light of the DNA, that two strands being complementary if separated by heating come together when appropriate conditions are provided. Very, very important line. What are they trying to say? That when I heat kill the strain, okay, when I heat kill S strain, at the first situation, that first instant, the heat kill S strain killed the mice. Okay, the mice died. But at the second instant, when I gave some more time with the help of R strain, what happened? The two strands which got separated during the heat came together. It came together, and that coming together caused the mice to die. So some students might have question, ma'am, but in the first instant S strain killed the mice, no? In the second instant, what will happen? Yes or no? That kind of questions can come. That is because of this, students, because in the first instant the strands would have got separated and some properties of it would have led the mice to die. But the second instant what has happened? We gave some time, no? So the strands came back together and they became very strong. Further, this presence or absence of that oxygen in the DNA makes it more stable, and because of the presence of that oxygen, RNA is very, very reactive. It becomes a reactive group and makes the RNA liable and easily degradable. This is the reason why RNA is not our genetic material, because the presence of that extra OH group, right? Presence of that extra oxygen group, that is OH group, in the RNA makes this RNA very, very reactive and easily degradable. Do we need someone who is not strong enough, okay, who cannot take up any changes in the body? What if sometimes heat is more? What if sometimes, temperature is different? That time, imagine our genetic material just getting melted down. We cannot survive like that, right? So it got easily degradable. That's the reason. 1 second. Okay, it was reactive, easily degradable. These reasons made RNA not be the genetic material in almost all the organisms. Okay, now RNA is also now known to be catalytic, hence reactive. Therefore, DNA is chemically less reactive. This could be because of the absence of, okay, could be because of the absence of oxygen, and structurally more stable compared to RNA, mainly because of the presence of thymine, okay, replaced instead of uracil. Thymine instead of uracil. Clear? Okay, great. Okay, this is what it is. Okay, thymine instead of uracil. Yes. Yes, they will get to know when they get a notice, right? Yes, okay, done. Thymine instead of uracil. Done. All of you made note. It will be great if you make note. Yes, okay, done. Fine. Now, students, look here. In fact, the presence of thymine at the place of uracil also confers additional stability to the DNA. Okay. Now, yes, detailed discussion about this requires understanding of the process of repair in DNA, and you will study this in higher classes. Okay. So here, the criteria. First criteria is replication. Both of them can replicate. Second criteria is stable, chemically and structurally stable. RNA is not chemically or structurally stable, so here RNA will fail. Next one is mutation. Both of them can mutate, but the problem is RNA will mutate at the faster rate. This is again a negative advantage. RNA will mutate at the faster rate. Okay. Consequently, viruses having RNA genome and having shorter lifespan mutate and evolve faster. Okay. Virus need to mutate faster. That is the reason why so fast virus, virulence, that means variants of virus keep coming, is because in virus RNA has to mutate faster so that they do not, they have to survive, right? They have to survive any kind of circumstances. So that's why RNA is a good genetic material for virus, not for us. Okay. Now RNA can directly code for synthesis of protein, hence can easily express the character. So this is a good point of RNA. They can easily code for protein because mRNA gets converted into protein, right? mRNA gets converted into protein. And because of this, they have the Mendelian characteristics. That means they can inherit, right? They can inherit whatever it is. Inherit, okay? They can inherit. DNA, however, is dependent on RNA for synthesis of protein. The protein synthesizing machinery have evolved around RNA. The above discussion indicate that both RNA and DNA can function as genetic material. Okay, they both can function as genetic material, but on a bigger scale, when we check out of all the organisms, DNA is the best genetic material. Okay, DNA can be the best genetic material or the most applicable genetic material. Why? Because of many other points. Clear? Done. Okay. Now, DNA replication. Okay, are you all ready for DNA replication? Okay, students, DNA replication, I would like to explain you in first making you understand different types of replication, and then I will tell you about the rest. Okay? Yes, different types of replication, and then I'll tell you about the rest. So, there are actually three types of replication. Okay, DNA replication is of three types. Three types. That means three different, not DNA replication, I would say replication is of three types. DNA does by only one form. Three types are there. Okay, let's write the names, students. First one is conservative. First one is conservative. Second one is semi-conservative. Okay, third one is dispersive. These three are the types of replication. In this, now our task is to find how does DNA replicate. Okay, because we told DNA will replicate, and that is why DNA is a genetic material. So this was ready to form its replica. Right, it is ready to form its replica. So now we are waiting for understanding what type of replication are they doing. Okay, so conservative, right, conservative. Okay, conservative replication, students. So three types of replication: conservative, semi-conservative, and dispersive. First, I will show you conservative just with colors. I'm going to teach you this entire thing with just colors. Okay, so let's draw parent DNA. Students, this is the parent DNA. Okay, who are they? Parent DNA. Parent DNA, template DNA. In some places, you can also see template DNA. Now they are going to undergo, types conservative replication. Now what is going to happen? Each of the strand, that means each of the parent DNA, so let's label it as A and B, A strand, okay, and B strand. These both are going to act as templates, right, students? Like how, like I'm telling you, go take Xerox. I'm telling you, go take Xerox. You'll be like, okay, ma'am, we will go take Xerox. Now, I will give you a copy to take Xerox, right? I'll give you a copy to take Xerox. This people will act like a Xerox copy. Is the main copy. The teacher's copy, we tell, no? Teacher's copy, I will give you one copy. You will go and take Xerox of that copy. Simple. So, here, this two will act like template and allow for the formation of the complementary strands. These two newly formed strands, right? Let's label it as G and H. Let's label it as G and H. This G and H will become the daughter DNAs. Okay, newly formed daughter DNA. Okay, daughter strands. Okay, you can call it simple way, daughter strands. This is one step of it. Okay. Now what are they going to do? I'm going to erase this part, students. I'm going to erase this part because this is understood, right? Okay. Now, students, what is going to happen? Parents will go back. Okay. The step will, if I continue, I don't think you'll be able to see, no? You'll be able to see? No, you'll not be able to see. Okay. So, students, what is going to happen? After this step, right? After they have replicated, what is happening? Parents will come back to their original. Parents will come back. Okay. Parents will come back. And this newly formed, okay, parents will come back and the daughters also will come back. Daughters will come back, parent will come back. This is called as conservative. That means they actually help in the template formation. Okay, they are actually the template. They will help in the formation of the new daughter DNA. But after replication happens, they will come back to their original places. That means the daughter DNAs will come back. A and B will come back. At the same time, G and H also will come back and bind with each other. This is conservative. Okay, this is conservative. Clear? Now, next one we are going to understand is semi-conservative. Okay, again we are going to understand with the help of colors. Let's keep the same thing. One second. Let's also I cannot draw entire box, but at least I can draw its difference. Okay. Next is semi-conservative. Again, let's draw parent DNA. Parent DNA. Again, A and B. Everything remains the same. Okay. Continue. They will also help in the formation of the template. Template A, B. Okay. Now the daughters will be formed. Right. Daughters will be formed. 1 and 2. Now look at it after replication, right? When they are doing replication, they will not go back. They will not go back to each other. That means what, students? This is G, this is H. They will bind with the parent DNA itself. They will bind with the parent DNA itself. This is called as semi-conservative. Can you see? Half of conservative, no? Here, till here, conservative and semi-conservative steps are same. Only here, what is going to happen? A and G will bind, B and H will bind. That's all. Simple. Okay? A and G will bind and B and H will bind, forming this as a semi-conservative. Okay? Semi-conservative method. Okay, this is how our DNA is going to replicate, but we do not know. We are still finding out. We are still finding out if this is how our DNA is actually going to replicate with the help of an experiment. Okay, with the help of an experiment. Now this is semi-conservative. This becomes conservative. Okay, this becomes conservative. That means A to B, and G to H. Now next one, students. Next one. Yes. Next one is your dispersive. Again, dispersive also, let's show it with colors. I don't think, if I don't move, I don't think it will be seen, no, students? Yes or no? 1 second, let me just check on the screen. Yeah, I'll do it on the top. Okay, I'll do it on the top. Yes. Now I'll do the next one on the top, that is your dispersive. Look at here, students. Dispersive, parent. Okay. Both the parents are there. Okay. What is going to happen is basically fragments will be formed. Okay. Fragments will be formed. That means fragments will be formed. That is, how do I mean? Like this. This is parent. From there, daughter strands will be formed. Got it? Like this, fragments will lead to the formation. Okay, this is what is called as dispersive method. Dispersive method. That means fragments will give rise to the daughter DNAs. Okay, this is dispersive method. Now out of this is the method our DNA will be choosing. Okay, DNA will be choosing this method, but how do we know it? We have to prove it with the help of an experiment. We have to prove it with the help of a experiment. So simple. Here what happened? Two parents are there. Each parent will act like a template. So I can call this as template. Okay, each of the parent will act as template and form copies called as daughter DNAs, right? These are called as daughter DNA. Daughter DNA. So here we have template, here we have daughter DNA. So what is going to happen? After they replicate, they will come back to their original position and they will all bind with each other. Parents will come back to their original position, daughters also will come back originally, and they will be happy. Now here what is going to happen? This is your semi-conservative. Parent is there, parent is acting like a template. Parent is acting like a template, forming the new daughter DNA. Forming the new daughter DNA. Now what is going to happen? They both will not come back. They both will not come back. They both only will bind. Parent and the daughter will bind, forming a semi-conservative method. That means, students, basically simple, they will form a hybrid. Our DNA will form a hybrid during replication. Then we have dispersive method, where a small fragment of the parent will help in the formation of the daughter DNA. Daughter DNA. These are the 3 types of replication. Okay, done. Now let's see how did they prove that our DNA will replicate by semi-conservative method. So here what is happening, students? First, what did they do? They took E. coli. They took E. coli. Okay, clear? There is actually one hindrance there, no? I don't know what is that actually. There is one, I don't know, I can see something. 1 second. This should be fine now. Okay, let's wait until the video load till there. Yes, okay, fine. So students, what is happening here? They took E. coli. Why did they take E. coli? Because E. coli replicates, okay, E. coli replicates every 20 minutes. E. coli replicates every 20 minutes. It duplicates every 18 minutes. It replicates every 20 minutes. Okay. Now, because of this, they took E. coli. This can come as a PYQ question. So, please make a note. Why did they choose E. coli and not anything else? Is because it replicates every 20 minutes. Okay. It replicates every 20 minutes. Okay. Little more I have to move, I guess. Yes. So, it will replicate every 20 minutes. Now, what did they do, students? Basically, they grew this E. coli cells in an N15 medium. Okay, they grew E. coli cells in an N15 medium, which was a heavy medium. Okay, and then after that, they grew this on an N14 medium. They grew it on a N14 medium, which was a light medium. Right, these are the 2 things. Now, what did they do? Okay, first they grew it in the N15 medium. So, as expected, they got 2 strands which were N15. This is the parent. Okay, N15, N15. First, if you grow E. coli in N15 medium, that is your nitrogen medium, this is actually your nitrogen. Okay, nitrogen medium. What is going to happen, students? In the first instant, we are definitely going to get both heavy strands, right? See, during centrifugation, they got a band which was N15. Simple. Again, after 20 minutes, they waited. That time, what did they do? After this, they dipped it in N14 medium. We need to see if our DNA will replicate in hybrid pattern. They dipped it or they left it grow in the N14 medium. Okay, after this, they let it grow on the N14 medium. They noticed that the DNA actually replicates in semi-conservative manner. Why? Because semi-conservative manner. Why? Because they noticed that one strand was N14 and the other strand was N15. Because if DNA was doing conservative, then we would have still got the multiplication. We would have got, instead of, see, from 1 we got 2 copies, right? The second, both the copies would have been N14 itself. But here what happened? Here we got a hybrid. Here we got a hybrid. This proves that our DNA replicates with semi-conservative replication. Clear? Semi-conservative replication. Now, students, understand. Again, 40 minutes they let it grow. So, 20 minutes, 40 minutes, 60 minutes, 1 hour they let it. They noticed that in the generation 2, they got both, like they got light and hybrid. How did they get in generation 2? They got light DNA plus hybrid DNA. Why did they get light DNA and not heavy DNA? Simple, students. They got light DNA because it was kept for longer time on the light DNA. Yes or no? Longer time was kept in the light DNA. Here you can see after N15 was formed, here onwards it is continuously kept in the N14 medium. No more in the N15 medium. So they got 2 strands of light DNA because light DNA, it was kept, that means in N14 they kept it, right? They kept it longer time in the N14 medium. Okay, hybrid DNA was observed. Here you can see 2 bands, you can see. Okay, 2 bands you can see. Now they use something called as your cesium chloride. This acts like a gradient, right? to give the difference. Okay, this will give the difference in the band difference. Okay, clear? Easy? Easy? Yes, okay. Now let's see there. They grew first E. coli in a medium containing NH4Cl. Okay, NH4Cl 15. This is the heavier one. This is the heavier particle. Heavier particle. Okay, it's heavy isotope of nitrogen. Simple, students, it is an isotope of hydrogen, heavy isotope of nitrogen, and as the only nitrogen source for many generation. Okay, so N15 was like a source of nitrogen for many generation. The result was that N15 was incorporated into the newly synthesized DNA as well as other nitrogen containing compounds. Simple, if you keep N15 for longer time, we would have got N15 itself on the DNA. Now, this heavy DNA molecule could be distinguished from normal DNA by centrifugation in cesium chloride density gradient. Understood? By centrifugating in cesium chloride density gradient. Please note that N15 is not a radioactive isotope and it can be separated from N14. Okay, it is not a radioactive isotope and it can be separated. They're basically just isotopes. They're not radioactive isotopes. Okay, based on densities, we can easily separate N15 from N14. Then what do? They transfer the cells into a medium with normal N14 NH4Cl and took samples at various time intervals as the cells multiplied and extracted the DNA that remained as double-stranded helix. The various samples were separated independently on the cesium chloride to measure the density of DNA. So in the first instance, they are basically telling E. coli was cultivated in N15 for a very longer time, because of which the newly formed DNA, right, the newly formed DNA was incorporated and we got N15 strands. Okay, after this what happened, students? After this, they kept it in the N14 medium, right? They kept it in the N14 medium and they noticed that we got a strand of N14 and we got N15. We got hybrid. Okay, after the replication on the newly synthesized one. Okay, newly synthesized DNA gave us both the hybrids. It gave us hybrids. Yes or no? So this is what has happened. Okay, this is what has happened. This newly synthesized N14, N15. After that, for a longer time, it was incorporated into the N14. Okay, for a very longer time, it was incorporated into the N14. So what happened at the generation 2, right? Generation 2, we noticed that there were two strands, or like 25% of light and 75% of hybrid. Okay, we noticed that there were only two types of strand. One is the light DNA and one is the hybrid DNA. Okay, this is what it is. Now if they reverse, that means instead of N15, if they had used N14, at the last situation you would have got heavy DNA. You would have got heavy DNA with hybrid DNA. You would have got heavy DNA with hybrid DNA. Simple. Okay, if this was reversed, instead of N15, if N14 was used and the same thing was continued, here we would have got one heavy DNA and hybrid DNA. Simple. That's all, students. Easy. Easy. Okay. Yes. Okay. Great. Yes. Fine. Fine for all of you. Yes, great. Okay. Done. Okay. Yes? Okay, great. Fine. Now, students, this much is clear. Enzyme machinery, we will start. We will take a kutty break and come back and then we will start. Okay? All of you take a kutty break because breaks are necessary. It will refresh your brain. Yes? Breaks are very, very necessary. Yes. Done. 1 second. Okay, fine. Let's take a break. What is the time? 23. Last time also we took break at the same time now. So we will resume back at 12:35. Okay, we'll resume back at 12:35. Clear? In case you people want to take a have breakfast or something, you can have and come, or lunch. Okay, I don't know what you people will have, but just yeah, you can have and come. My throat is paining, so we will take a small break. Okay. I am back. Are you all back? Okay, let's start the class, students. I hope all of you are back. Yes, I can see some of you are back. Very good. Okay. Yes. Okay, so what are we trying to learn here? We are trying to learn enzyme machineries, right? Okay, students, now we understood replication, right? We understood DNA is doing this. Sorry. DNA is doing this, DNA is doing that. We understood this much. Now we have to understand. We understood DNA is going to, replicate with the help of semi-conservative method. Semi-conservative method. Yes or no? Now, this semi-conservative method, when we talk about, we did not talk about how it is going to happen, right? We just told that, yes, semi-conservative method is happening, but we did not tell how this double-stranded DNA is getting separated, how are they acting like templates for each other, all this now we have to understand. So, for enzyme machinery, I would say there are 4 enzymes, okay, 4 enzymes. First one is your DNA. No, let's go in order itself so it'll be easy for you people. First, let's do helicase. Who is that? Helicase. He is the person who unwinds. He is the unwinding enzyme. Who is he? Unwinding enzyme. Okay. Now, what am I telling? What is Gopika ma'am trying to say? Gopika ma'am is trying to say this is how your DNA is and this is how your 2 strands are. Now, we need to unwind it. If I don't unwind it, how am I going to form the template? Yes or no? How am I going to form the template, right? So, for this, helicase will help me unwind my DNA like this, right? Like this. It will form like this. Have you seen structures like this on your textbook? It will form like this. Correct? So, unwinding enzyme is helicase. So, helicase will remove the bonds, remove the loops and make this twisted strand into a straight strand. Okay? Now, we call this structure as a replication fork. What do we call it as? Replication fork. It looks like a fork, no? Even though it is 3, it looks like a fork. So, it is called as replication fork. Done. This much clear. Now, the next thing is, I want to start my replication. Okay, now that unwinding has happened, I've got my template strands. No problem. My tension is over. Let me label it 5' to 3', 3' to 5'. Okay, I've got my strands also. Now the tension is I want to start my replication. How do I start? So for the replication to start, right, we need to find a spot, no? We need to find a starting point. So replication will start at origin of replication or your ORI. Origin of replication or your ORI. Okay. This ORI spot, students, usually you should understand this ORI spot will be in the AT region. That means adenine and thymine region. Yesterday I told you these are 2 bonds, right? And because of that it is easy to break. So origin of replication will be mostly at the A and T region. Why? Because bonds are weak to break. G and C has 3 hydrogen bonds, more stronger, difficult to break. So A and T will be the bond that we use to break. Okay, clear? Yes. A and T region, that is called as ORI, origin of replication. This much is clear, which will initiate. This is where initiation will happen. That means where initiate replication. Initiate replication. Done. Now we have to start, we need to form the next strand. Yes, okay ma'am, you have separated it, I understood. You told me that it is going to start at origin of replication and it will be in the AT site because the bonds are easy to break. Ma'am, but how will you form the daughter DNA now that you have separated the strand? That means the teacher copy is ready. Now we need to make the daughters, right? For that, what will I do? For that, I am going to take the help of the next, oh sorry, I'm going to take the help of the next person, that is your DNA polymerase. To form new DNA, I will need DNA polymerase, right? DNA polymerase. DNA polymerase is ready to come, but he has some condition. DNA polymerase has some condition. He's like, I will only work on a strand which is single. Okay, I will only work on a strand which is single. I will not work on double strands. I'll work only on a strand which is single. Second thing, I will only work on a strand, okay? Yes, okay. I will only work on a strand which is from the 5' to 3' end. I will only work on a strand which will go from 5' to 3' end. I will only work on this strand. I will not work from 3' to 5'. We were like, okay, fine, it's okay, you come and do the work. No, we're like, we call people to do the work. They'll let some condition, I want biryani in the lunch, I want to, I want to take a 1 hour nap. We agree to all of this because we want the work to get done. So DNA polymerase will start its work from 5' to 3'. But DNA polymerase have a problem. It does not know where to start. Yes, origin of replication it will start, but it does not know, it needs someone to lead it. Okay, that person is going to be your primase. Okay, primase is the person who is going to lead your DNA polymerase. DNA polymerase will be led by the primase. Okay, primase also came. Students, you can form the strand in any ways. Okay, from here if you add the primase also it will continue. From here if you add also it will continue. So imagine we add the primase here, DNA polymerase will start doing its work and it will form a template strand. Okay, it formed a template strand. Okay, I will draw this a little more down, I guess. Yes, okay. Now let's draw the next part of the strand. Okay, this is 3' to 5'. Yes. So this strand which we formed with the help of RNA polymerase, this strand, the blue one, this will be called as the continuous strand because you can see no breaks are there. At one shot he formed the strand, right? Continuous strand. Now the problem comes. One more strand we have. Okay, one more strand we have. That strand, problem is that the strand is from 3' to 5'. What is DNA polymerase condition? I will only work on a strand which is 5' to 3'. Now problem has come. Now what do we do? Then primase will be like, anyway this fellow does not know from where to start. I will do one thing. I will start doing it reverse. That means I will still do it from 5' to 3' but in a reverse direction. So primase will go attach itself here. DNA polymerase will start forming. DNA polymerase will start forming. Because it is reverse, it needs more effort. So again primase has to come and attach. Okay. Again polymerase will form. So students, basically this will be a discontinuous process because it is in reverse pattern. No, it will be a completely discontinuous process, right? So it will form like this. Finally, we will get the strand. Now this strand is called as discontinuous strand. But the most famous name, okay, more than discontinuous strand, the most famous name is Okazaki fragments. Okazaki fragments. Okazaki fragments. Okazaki fragments are actually these fragments, right? Because this DNA polymerase is not ready to do anything. What can we do? What can we do? So this DNA polymerase is like, okay, fine. I am not going to work from 3' to 5'. So primase will be like, don't worry, I will do one thing. I will start doing in the reverse pattern and somehow we will form the fragment and that is called as Okazaki fragments. Continuous strand is also called, okay, continuous strand is also called as the leading strand. It is also called the leading strand. and discontinuous strand or your Okazaki this is called as your lagging strand. Now work got over. Okay, work got over. Then DNA polymerase. Now class will get over. I'll tell students, please do like, please do comment if it's good. You people don't do, no? That means after work is over, who cares? My work is over. Same way, DNA polymerase work will get over, and DNA polymerase is like, I don't want any more primers. Remove all the primers. Okay? I don't want any more primers. Remove all the primers. Then we need to fill this gap. No, we need it as a fragment. I don't want it like a fragment. I need it like a strand. So then one more person will come, that is called as your, okay, that is here we will write it. That is called as your ligase. Ligase is also called as your gluing enzyme. It's also called as your gluing enzyme, which will actually glue all these people together. Okay, it will actually glue all the fragments together. But student, space is there, so that means we need to form some nucleotides. So that DNA polymerase only will do, but the gluing part, right? Gluing part will be done by your ligase. Finally, we will get 2 fragments. Okay, finally we will get 2 fragments. So this will glue with the help of DNA polymerase. DNA polymerase will fill the gaps. Okay, DNA polymerase will fill the gaps. And this sticking job will be done by ligase. So finally we form strands. Okay, finally we formed, what did we form? We formed a parent DNA and we formed a daughter DNA. Okay, again we formed a parent DNA and we formed a daughter DNA. Yes or no? This is what you see here. Yes. So one is from 5' to 3', and this is from 3' to 5'. So this strand, 3' to 5', this is a continuous strand. Here 3' to 5'. This one is a discontinuous strand, but then yeah, we have completed the strand, but still. Okay, this is from 3' to 5'. This is from 5' to 3'. So by this method, we got our semi-conservative. By semi-conservative method, we did replication of DNA. Okay, replication of DNA. Done. Okay, this is all your enzyme machinery. Yes or no? This is all your enzyme machinery. Okay, this is all about it. Clear? Very easy. So students, what is happening? First, unwinding is happening with the help of helicase. Unwinding enzyme, it will unwind this DNA helical structure to a replication fork. Okay, this is called as the replication fork. Let me label it. This is called the replication fork. Okay, this replication fork will have an ORI. Okay, origin of replication. It will be mostly an AT region because AT bonds are weak and it is easy to break. Correct? From there, what will happen? DNA polymerase will come to form new DNA strand or your daughter DNA. DNA polymerase has some condition. I will only work on a strand which is single. That means we need helicase help to make the strand single. Second thing, I will only work on a strand which is from 5' to 3'. 2 conditions also. We'll be like, okay, you do whatever you want. We will agree to the condition. He will come, start forming new strands. He will form strands easily, but the problem is he does not know where to start. That time who will come? Primase will come for help. Primase will attach itself, right, and form the strand. Now after that, primase will be like, okay, done. We formed a continuous strand. Now problem is no more there. We formed the continuous strand and right, like the problem is sorted. Okay, continuous strands will be formed, then discontinuous strands. Now second problem is DNA polymerase will not work from 3' to 5'. Problem came. So that time DNA polymerase will be like, okay, I will not work from 3' to 5'. Primase will be like, don't worry, I will do one thing. I will mislead this fellow. I will attach myself from the back side, right, from the reverse pattern, and we will make him form the strand. Because it was from the reverse pattern, the strands will not be formed easily. It will be in a discontinuous pattern. And because of this discontinuous patterns, what will happen? We call this as Okazaki fragments. Okay, the person who found it is Okazaki. So Okazaki fragments. And after one point, DNA polymerase will be like, come on, I don't want this primer's help. Just throw the primer out. So we will remove all the primer from both the strands, even from the strand. Okay, and we will glue all of this together with the help of your ligase. Okay, ligase is gluing, but new nucleotides are formed by DNA polymerase itself. Okay, by this, what did we get? We formed replication. We did replication with the help of semi-conservative method. So the enzymes that are involved, right? The enzymes that are involved starts with helicase, okay, which is also called as the unwinding enzyme. Students, with this name only you will be able to easily remember. Okay, this is the best way to study replication. It is usually complicated, but then now see how easily you will understand. Helicase. Second one is DNA polymerase. Okay, DNA polymerase, which will actually help in the formation, okay, formation of new strand. Then at the third, after the second, we have who? We will have primase, because this person does not know to go by itself. Primase will lead the DNA polymerase or guide DNA polymerase. And last we have ligase, which will help in the gluing. It's a gluing enzyme which will help in gluing the fragments. After we glue the fragments, what do we get? We get it into a continuous strand, right? After we glue all the fragments, it will look like a continuous strand. This is all about your enzyme machinery. Okay, then we have some more things, I'll write. Then we have ORI. Full form is origin of replication. Okay, origin of replication. Okay, where this entire thing will be initiated. Done. That's all. That is all your enzyme machinery. At the end, we will be getting replication done. That means we will form replica of the DNA, proving DNA to be the genetic material. Yes or no? Because if it's a genetic material, it has to form a replica, right? Yes, done. Fine. Look here, students. This is how it will look. See, you can see here what is happening. This is the leading strand. So new things are formed. Okay, DNA polymerase is moving like this. Can you see DNA polymerase moving like this and forming the leading strand? So helicase will slowly open up. Okay, helicase will not open up the entire 2.2 meters. It will not open up. It will slowly open whatever is necessary for the DNA polymerase. And here you can see lagging strand. Okay, again DNA polymerase, original DNA strand, you can see addition is happening slowly. Okay, but can you see this is going this way, this is coming in this way. Okay, this is how the primase will cheat on it, right? Primase will be like, don't worry, I will make him understand, right? So that is what is happening. Okay, these are free nucleotides which are waiting to get added. Okay, that's all simple. Now next process is transcription. Okay, so students, according to our central dogma, we have understood right now only we have understood one technique. Right now we have only understood DNA forming its own replica. That means by the process of replication. This is how much we have understood. In replication also we only understood that it is semi-conservative. Okay, and we proved it with Michelson and Stahl experiment. Okay, we proved it by Michelson and Stahl experiment. That's all. Okay, now the second topic, right? The second topic is DNA can form mRNA, and that process is called transcription. Yes or no? So DNA can form mRNA and that process is called transcription. Yes or no? Now we are going to learn about transcription. That is the topic for today. Okay, that is the topic for today. So what is basically happening? We have to form from DNA, we have to form mRNA. For that, we will need some enzymes. Okay, the first enzyme will be, students, definitely RNA polymerase. DNA dependent RNA polymerase, or you can just remember RNA polymerase, whatever is easy for you. In case in option DNA dependent RNA polymerase is asked, then you please go for it. Otherwise, RNA polymerase. Okay. So RNA polymerase we need because we are forming from DNA mRNA. Okay. Now what is happening? There are actually three, like we have a transcriptional unit. What do we have? A transcriptional unit. I'll tell you how it is. We have a transcriptional unit. What is a transcriptional unit? First person who is there is promoter. Okay. Second, we have structural gene. Do not worry, at the end of this class, this will be very, very clear for you. Terminator. These three people are there. Now, from the term itself, you are understanding promoter. Something it will promote, right? So, in the promoter region, students, this is the transcriptional unit. Promoter, structural gene, terminator. Now, we are going to understand transcriptional processes, which are the processes involved in transcription. In the promoter region, initiation will happen. Right? Promoter, we can understand. Initiation is going to happen. Structural gene region, elongation will happen. This is the process. Okay, elongation will happen. Terminator, termination will happen. Yes or no? Termination will happen. Simple. Okay. So these are the unit. This is the unit. This is the process. Okay. This is the process that takes place in your transcription. Now let's go one by one. Let's go one by one. Okay, all of you look here. So this is what we understood. Now what is going to happen in students? First we will try to understand structural gene. Can I draw a line? No, right? No, I cannot. Okay. So students, first thing is structural gene. Let's talk about who is this person. Promoter, you understood. Like one word. It is ma'am initiating or promoting something. Structural gene, students. Do I need to make the entire 2.2 meters of DNA into mRNA? No, ma'am, I do not need to do that, right? Why will I take 2.2 meter DNA and convert it to mRNA, right? It is crazy. No need that. So, I will take a small region. I will take a small region and I will make that small region into mRNA. Now, that small region is called as my structural gene. That small region is called my structural gene. Clear? Okay, so entire 2.2 meters I will not convert it into mRNA. I will take a small portion of the DNA and that portion is called the structural gene. So if you notice, all of you would have seen this diagram where you would have seen something like this, right? In the textbook also it is there. Like this. I think you people will not be able to see because I am sitting. 1 second, we'll draw it in the next, this one. But yeah, I will try to draw it here above, little smaller. So students, you would have seen like this. Right? Right, you would have seen something like this. Here you would have seen this. This is your structural gene region, no? The small region which is going to form the mRNA. Okay? This is it. Now, we have promoter, fine. In the initiation, what is going to happen? Initiation is going to happen. That means we are going to initiate the reaction. Simple as that. Right? We are going to initiate the reaction. So, in the initiation reaction, we need someone, no, to tell this RNA polymerase, oh bro, you're coming. Okay, let's do one thing. I have to open the gate for you, right? For that, some person should be there, no? That initiation will happen because of sigma factor. Initiation will happen because of sigma factor. That means RNA polymerase will be coming, right? RNA polymerase will bind with sigma factor. Who it will bind? RNA polymerase. Important, RNA polymerase will bind with sigma factor and help or continue your initiation. Only if it binds with sigma factor will it be able to do initiation. Clear? Okay, done. Structural gene region is the region where elongation happens. That means where your mRNA will be actually formed. Then termination. Again, RNA polymerase have no bell and break. RNA polymerase will be like, I don't know, I just started the reaction, I bound with sigma factor, I started initiation. Now, RNA polymerase does not know where to stop, right? So, polymerase will go and attach to rho factor, which will actually help in the termination. Clear? So, all you need to remember is the transcriptional unit, processes, and you should know that in the initiation, that means in the promoter region, we have sigma factor, and in the terminator region, we have rho factor. Okay? Done? Now, look here in your textbook, this diagram is given. So, students understand, two things are there. Okay? For transcription to happen, we will always use only one strand. We will always use only one strand. You'll be like, why? Okay, and that strand will be called as the template strand. That means the strand which is going to form mRNA, right? That strand is called template strand. And one more strand will be, we all know DNA is two strands, right? So one strand, and let's say red, one more strand. Okay, this one more strand, which we are not going to bother about, is actually the complement or coding strand. Sorry, it is the coding strand, which will not take part in the transcription. You'll be like, ma'am, why? Simple, students. I have two strands. Now say A, G, T, C, A, G. This is my strand number 1. Let's put it in same colors. Okay, this above this I have, let's say T, A and T will bind, C, A, G, T, C. Okay, this is my strand number 1, S1, S2. Now, this strand is going to form an mRNA. This strand is going to form my mRNA. This is my template strand. This is actually my template strand, and this becomes my coding strand. Now, both are going to form mRNA. Okay. Let's make mRNA from both of them. Simple, thymine will be replaced by uracil. So, UG, no? Yeah. UG, T, C, A, G. Yes or no? Thymine will be replaced by uracil. Wait, 1 second. I think I wrote here T, no? It also will be U. 1 second, students. 1 second. Ulta. We have to write it ulta. Okay, here we have A and here you will write U. Simple. Okay, this is the mRNA which I got from this template strand. Complementary, right? Okay, mRNA done. Here also let's draw an mRNA. Let's use another color. Okay, A will be T, no? So U, G, C, T, A, C, G, A, U, G, C. These are the mRNAs. Now this is my mRNA 2. This is my mRNA 1. This is my mRNA. Just look at this. Don't you think these both are complementary? A will bind with U, G will bind with C, U will bind with A, C will... They're complementary to each other. So what will happen? mRNA 1 and mRNA 2 will be complementary to each other and they will bind. Now to avoid this, I will only take one strand of the DNA for the transcription. Because why do I want such complications? Because I know that according to central dogma, one DNA will give rise to one mRNA. This is only about eukaryotes, okay? This is about eukaryotes, not about prokaryotes, okay? Prokaryotes can do multiple. One DNA will form one mRNA. That one mRNA will code for one protein. Will form one protein. This is the general central dogma. Now, if I take two DNA, two mRNA, still both this mRNA will bind and do what? Right? So, I will always follow the central dogma, that is one DNA, or say one DNA will code for one DNA will form one protein through the process of central dogma. Clear? So, this is the reason why we are not taking two strands of the DNA. We are only using one strand. And the strand we use is called the template strand, and the strand we do not use is the coding strand. But you have to remember one thing about the coding strand. Coding strand and the mRNA. See, if you look here, students, this one. Right? Coding strand and the formed mRNA will be the same. What did I say? coding strand AGTCAG. Look here, ACUCAG. Same thing, no? Only thing thymine is replaced by uracil. Thymine is replaced by uracil. So the coding strand and the mRNA formed will have the same nucleotide, except that thymine will be replaced by uracil. That's the only thing. Okay. Now look here, you have a promoter region, you have a terminator region. In the promoter region, you will have a sigma factor, which will favor the reaction, okay, favor the process, and here you will have the rho factor. It will form the rho factor. Okay. Here we have structural gene, and here. Now look here, students, what is happening? So RNA polymerase is coming. This fellow is the one who has to form the mRNA. This is the fellow who has to form mRNA. So he is coming. Okay. So we are using only one strand. Okay. He's coming, and this region, right? This region is called the structural gene region. That means the region which will only form the mRNA. Everything will not form mRNA. Okay. Now sigma factor, you can see RNA polymerase. You can see they both will bind, and they will come and see one mRNA came out. Aram se one RNA came out. Now rho factor. Now sigma factor went because RNA we got. That process is called elongation. That means formation of your mRNA. Elongation. Okay. Now termination. RNA polymerase have no control, right? RNA polymerase will keep going like this. No, that time rho factor will bind with RNA polymerase and tell stop, enough of making the RNA. Now this much is done. Okay, this is all about your transcription. If you want, we will quickly revise it. So students, transcription basically happens in three regions, that is your promoter, structural gene, and terminator region. In the promoter region, initiation will happen with the help of sigma factor. RNA polymerase will bind with the sigma factor and continue the process. Okay, 1 second, let me see if I'm yes. Okay. Yes, okay, it's clear only, no? I thought it was too small. Okay, so students, what is happening? Basically, in the in the initiation, sigma factor will be there. It will bind with RNA polymerase and help for the initiation of the reaction. Then there is a structural gene region, which is the actual region that will help in the formation of the mRNA, because the entire 2.2 meters of DNA forming mRNA does not make sense, right? That region is called the elongation region. There elongation will happen. Then we have termination, where the reaction will terminate. That means RNA polymerase will bind with the rho factor and it will stop the reaction, right? Now, why are we taking only one strand? Why are we not taking two strands of the DNA? Is because according to our central dogma, one DNA will form one protein. This is for sure. Okay. Now, for that, or this one small region will form one protein. For that to happen, I need to take only one DNA strand, because if I take two DNA strands, they will form mRNA which are complementary to each other and they will bind with each other. Now, two mRNA cannot bind with the protein, right? Complementary mRNAs cannot bind with the protein, right? Because tRNA will read only one of them. So, to avoid this, I will be only using one strand, and that strand is called the template strand. The strand which I'm not using, usually the 5' to 3' that will be called the coding strand. This coding strand and the formed mRNA will be same. The nucleotide will be same, just that thymine will be replaced by uracil. Simple. Now, we got the mRNA. Okay, happy. Now, we have to understand something a little complicated, but I will make sure that I'll make it very easy for you. So in gene, or there is a word mentioned in your NCERT called cistron. Okay, there is something called as cistron. Cistron means a small region or a sequence of a region or a sequence of DNA. A small region or a sequence or a section, anything, any of the words you can use. Okay, sequence, section of a DNA. You'll be like, ma'am, why are you telling us about this, right? So since cistron is mentioned there in your textbook, and there are two types mentioned, monocistron and polycistron. Monocistron and polycistron. What is monocistron and polycistron? Okay. Our eukaryotic gene. What is our eukaryotic gene? Is little complicated. Eukaryotic gene is little complicated. We have something called as the split gene. What did I say? Split gene. I'll tell you why it is. Okay, all if you look here, this is how our eukaryotic gene will be. That means we have some portions, okay, we have some coding and non-coding sequence. Okay, eukaryotic will have some coding and non-coding sequences. Some coding and non-coding sequences. Coding sequences, this coding sequences are called exons and non-coding sequences are called introns. Okay, because the genes are like this, right? Because the genes are like this, the formed mRNA also will face the same problem. Yes or no? Structural gene is the one that forms mRNA. If structural gene is a split gene, that means the gene itself has some coding and non-coding sequence, then the mRNA formed also will have this problem. Yes or no? Okay. Now the formed mRNA, we have to make sure that they do not have any non-coding sequences, because if they have non-coding sequences, they will not code for protein. Then our entire central dogma will fail. So for that, students, we do something called as splicing. We could do something called as splicing. Okay, that means splicing is the process by which we removal of introns. Simple words, you can tell removal of introns, or you can tell removal of non-coding sequences. Non-coding sequences. Okay, so by the help of splicing, we will be able to form a gene which has only exons, or like the mRNA with only exons. That means mRNA with only coding sequences. Okay, coding sequences. This process is called as RNA processing. What is it called as? RNA processing. This is not there in your this is not there in your prokaryotes. It is only there in eukaryotes. RNA processing. So RNA processing is only because in our eukaryotes we have a split gene, and we can also say, okay students, you in your textbook there is also hnRNA, right? If you notice hnRNA, okay, there is your heterozygous heteronuclear RNA. Okay, heteronuclear RNA. hnRNA called as your heteronuclear RNA. Right, different. Hetero means different, mixed. Right. So with the help of splicing, we will remove all the introns or the non-coding sequence, and we will get an mRNA which has completely coding sequence. This mRNA will be capable for coding for protein. Now the catch is, I need to make sure that I'm protecting this from any kind of restriction endonuclease or any kind of unnecessary attachments. So for that, what I will do, I am going to take this mRNA and I'm going to add a cap. And also students, this capping and tailing is very important to know the ends, 5' to 3', which end, right? So I'm going to put a cap, and at the same time I'm also going to put a tail. A capping and tailing will be done. Okay, so capping will be done with the help of methyl, what is mentioned in your textbook, okay? Methyl guanosine triphosphate, and tailing will be done with the help of poly-A tail. Poly-A tail. With this we will do tailing. What do I mean? I basically mean that I am also getting to know the ends of the DNA. So I will say 5' and 3'. That's all. Okay, I'm also getting the ends of the DNA. Okay. So on the capping end, we will get the 5'. On the tailing end, you will get the 3'. So this way, what are we doing? We are basically protecting this mRNA from not getting affected by a restriction endonuclease. At the same time, we also have a head and tail for it. So we are protecting it. Okay. Now when we talk about polycistronic, okay, here let's write here Polycystonic. What is this? I told you cystron is a section or a sequence of a DNA. Polycystonic, students, is seen in prokaryotes. So you're going to remember PP. It is seen in prokaryotes. That means according to the central dogma, one gene, right? One gene can code for multiple. Here, students, monocystonic, one gene can only code for one protein. Okay, this will only code for one protein. That is also here. Here, what is happening, students? Genes are there. It will all code for multiple proteins. No problem. No problem at all. It will code for protein, protein, protein. It will code for multiple proteins, and they do not have no split gene. They do not have split gene, nor they have any, introns. Like no, they do not have any introns or non-coding sequences. No non-coding sequences will affect their continuation of your translation. Okay, this is only present in our eukaryotes because we have little complicated. Okay, now this poly A tail addition is also called as polyadenylation. This capping and tailing, no, this is called as polyadenylation. So this is the whole process. Clear or not? Okay, now we have finally formed a really good mRNA which can be utilized, right? Which can be utilized to form the protein. Problem sorted, right? We have formed an mRNA that will now code for protein, and that process will be called translation. And for that to happen, we need some genetic codes. Okay, we need some genetic codes which will be always in the form of triplets. This is the thing. Okay, polycystonic and monocystonic, I hope it's clear. Monocystonic, that means that section of DNA or region of DNA will only code for one protein. Here, polycystonic, different regions will code for different proteins, and they do not have any split genes, and because of that, they do not have to undergo. Very important, I'll write that also. They do not have to undergo. They do not. Okay. These ones do not have to undergo RNA processing. They do not have to undergo RNA processing. Simple. They do not have to undergo RNA processing. Why? Because they do not have split gene. Split gene, only when we have split gene, we have to undergo RNA processing. This RNA processing is the method by which we remove that means where splicing will happen, capping will happen. That entire process is called RNA processing. Okay. So exons and introns are the exons are the coding sequence. They are the coding. These are the non-coding. These are the non-coding sequence. We have to remove the introns with the help of a method called as splicing. After that, we'll add a cap and tail to the mRNA, and we'll properly get a good mRNA. So this is what is shown in your NCERT. If you look here, you can see there is strands of DNA. Okay, and then here you can see capping is done. This is methyl guanosine triphosphate. Introns are there. This introns will be removed with the help of RNA splicing and poly A tail. I'm so sorry, student. Throat is paining. Okay, poly A tail. Here you can see polyadenylation, capping is done, tailing is done, and this is how we get a newly formed mRNA, and we will also have 5' to 3'. Okay, starting only you can put 5' to 3', but also if you see some textbooks, they will say 5' to 3'. That is also done after, what is that? Capping and tailing. Okay. Next topic is genetic code. I will not go a lot into this, but just let's have an introduction. So George Gamvo was a physicist who argued that since there are only 4 bases, if they have to code for 20 amino acids, the code should constitute a combination of bases. Simple, students, don't break your head. I will explain it to you. Students, what are they trying to say? He's trying to say there is adenine, guanine, cytosine, thymine. These are the 4 bases. But I need to code for 20 amino acids. I need to code for 20 amino acids. He's like, I cannot individually use them. Now imagine I individually use them. 4:1. I will only be able to code for 4 amino acids. That means adenine will code for 1, guanine will code. Okay, wrong. Then he was like, there should be some combination. Then they were like, okay, let's do one thing, 4 power 2. Like, we'll keep 2 2 as a combination. Then they were like, okay, 4 is 4 into 4, which will give 16 amino acids I can code. Then what about the rest 4? Then they were like, okay, last option is 4 is to 3. That means 4 into 4 into 4, which is 64. We will be able to code for 64 amino acids. It's okay, 20 is the amino acid. Maybe future, if we find more amino acids, we will code for that, right? So finally, they made 64 codons. Now, what are codons basically? They are just codes that will be capable. That means for simple students, mRNA to protein. mRNA to protein. This mRNA will have some codons which will actually help us understand which protein should it code to. Now imagine valine is there, methionine is there, phenylalanine is there. Okay. Now many proteins are there. Now these proteins, leucine is there. Okay, many proteins are there. Now this mRNA will have a set of codons which are always in C3 triplets. They will always be in triplets. Now example AUG codes for methionine. This triplets will let us know that what, where, when, how it should code. That means if on an mRNA, if I see so many codons, AUG, UUU, UAG, UCG, okay, then CCU, all this, many codons are there, okay. Now this will code for one protein, 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 this will code for one protein. This is called as codons. That means these codons will be read with the help of who? With the help of tRNA plus ribosome. tRNA and ribosome will form, okay, tRNA and ribosome will form a unit that will help in the coding. And this tRNA will have two regions, okay, one amino acid acceptor site, and it will have, at the same time, it will also have an anticodon loop. It will have an anticodon loop. Okay, so is this clear? These are the codons. So how is this codon, students? Understand, there are 1, 2 and 3 rows. Okay, 3 rows are there. So here U, can you see here? U, C, can you see here? U, C. 1, so it will go like this. 1, 2, 3. In the 3 there are 4, right? 1, 2, 3, 4. So it will go 1, 2, 1, 1, 2, 2, 1, 2, 3, 1, 2, 4. Like that you can always do for entire thing. But these codons are not like, you don't have to by heart or anything. But you should know that there are 64 codons. Okay. And each of the codons will be always in triplet. And this will help in the formation of your polypeptide chain. This much if you know is more than enough. Some are there, that exceptions we will do polypeptide chain. Okay. Then there are some salient features of your DNA. Of your genetic code, which is mentioned in your textbook, right? Now, 1 second, students. I've written it here, but then it's just gone. Yes, 1 second. Okay, now you can see the codes. Here also, no, here it's not there. Okay. Yes. Students, look here. These are the salient features. Okay, salient features of the genetic code. So, what are the salient features of genetic code? The codons are always triplet. Okay, 61 codons are there. Okay, here it's very important. 61 codons are there. No, 64 codons are there. Out of which only 61 will code for proteins. Only 61 will code for some protein. Then what about the 3? Those 3 codons are called as nonsense codon, or stop codon. Okay. So out of 64 of them, 61 will code for protein and 3 of them will code or become stop codon or nonsense codon. Okay. Now, hence they function as stop codons. Some amino acids are coded by more than one codon, hence the code is degenerate. Okay, that means if you see UUU will code for phenylalanine. Again, if you see backside here, you can again see UUC also is coding for phenylalanine. Okay, that's all. That's all. Yeah, phenylalanine. So like that. See, oh sorry, sir. Yes. So UUC also is coding for phenylalanine. That means some amino acids are coded by more than one codon. Okay, the codon is read in mRNA in a contagious fashion. That means, students, I will not see. So this is my mRNA. I will not read AUG, UUU. I'll read AUG, UUU, UUC. I will read it continuously without any comma, full stop, semicolon, nothing. Just continuous pattern. Okay, in a continuous manner. Okay. Now, next, that's all right. It's read in a continuous manner. Then, students, here actually some more points are there. It is universal. We'll write it here. Yes. The codons are always universal. That means if UUU is coding for phenylalanine in bacteria, okay, even in humans also UUU will code for phenylalanine. Humans also, it will code the same. Simple. Okay, it is universal. No matter which small, big, whatever organism they are, the same codons will code for the same amino acid proteins. Okay, then AUG, some codons will have dual function. Like AUG will code for methionine, but it is also acts like a start codon. It is also a start codon. Okay, now if you look here, you can see which are the stop codons. Okay, UGA. Wait, I will write it here separately. UGA, UAA. Okay, these are all stop codons. So please make a note. UAG. These three are the stop codons. These three are the stop codons. Okay. So here, if you see same letters. Okay, students. UAG. Different combinations are made. So how is it made? This one. UAA. Okay. UAG, one combination, and then UGA. Okay, UGA. Three combinations. So A will come 2 times in the center, G will come 1 time in the center. Always remember this. Okay, if you remember that way, it will be easy for you to make the combinations. Okay, how will you remember? A comes 2 times in the center. Okay, A comes 2 times in the center, G comes 1 time in the center. UAA, UAG, UGA. That's all. Simple. Okay. That's all is the combination. This is how you'll remember. 2 times A in the center. Okay. And 1 time G in the center. Fine. This is done. Universal. Okay. Methionine. Now, insertion and deletion. I think we will do it next class. Okay. But I will just show you. I think we'll do it here only, students. It's easy. Okay. Ram. This is the textbook example. Ram has red cap. I'll explain you only with this. Ram has red cap. Now, okay, Ram has red cap. I decided to remove H from it. Okay, no, I'll remove S from it. Ram has red cap. What did I do? I basically, deletion number 1 happened. Okay, deletion happened. That is number 1. That is S I deleted. Okay, so now what is going to happen? Your tRNA only cares about the codon. It always reads in triplets. So 1 triplet, Ram makes sense. Hard. Okay, this 3 will become 1 codon. So this is 1. This is the second that tRNA will read. This is the third tRNA will read. AP will not be read. Okay, AP will not be read. Now the entire codon has gone wrong. Okay, because this will code for 1 protein, this will code for 1 protein, this will code for 1 protein. But is this the protein that it has to code? No, because here it has changed, right? S has gone. Okay, S got removed. Now again I decide. Okay, fine. I will do one thing. I will write Ram. Okay, I am again going to remove A. Okay, red cap. So whom did I remove? I removed A and S I removed. Okay. Now again tRNA will come. What will it do? It will code like this 1. It will code like this 2. it will code like this 3, and P will not be coded. P will continue to code with the next letters. Now what is happening? Again, the protein composition is different, because every time a code is changing, protein also will be different, right? Now what is happening, students? This will actually, see, now I cannot read it anymore, right? Same way how I cannot read it, even the tRNA will not be able to read it. tRNA also will not be able to read it, and that will lead to frame shift mutation. Like how we have a reading shift, no? Like Ram has red cap. Now I cannot read it. Ram held cap. I cannot read it. So how my reading shift also changed, that is also called as frame shift mutation. Okay, the frame has shifted, right? That frame shift mutation has happened. Okay, this is by with the help of deletion. Frame shift mutation happens because when some code gets deleted, code or one nucleotide, even one nucleotide changes also. Like students, how you have sickle cell anemia, right? Point mutation. Even one nucleotide changes also, it will cause a frame shift mutation. At the same time, imagine, okay, at the same time, imagine I'm writing here, okay, I will erase this. Yes. Now imagine at the same time I'm writing Ram has B red cap. I inserted a B here, right? I inserted a B here. I inserted a B here. Now what has happened? 1, okay, tRNA will start reading. 2, 3, 4. P is left alone. That means it will be reading in such a way that Ram, okay, has. Now with B I decided to add I also. So this will code for some, this will code for something, this will code for something, this will code for something. P will be continuing with the next person. Now again I added one more letter. What is going to happen? Ram has Bir, okay, E, D, C and AP will be left alone, correct? This will go for 1, 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 okay, and this 2 will be coding with next person. Here what has happened, student? Here what has happened? Insertion mutation will happen. Insertion mutation. That means by inserting something, I have changed the entire system. By inserting a nucleotide or a base, I think better than nucleotide is a base, because nucleotide is a wrong word. Okay, I'll add a base. Okay, inserting a base will change the entire, right, the entire codons. And the tRNA will start reading 3, 3. tRNA only knows one thing. tRNA, no matter what, it will always read as a triplet. It will always read as a triplet, and because it reads as a triplet, it does not know if it makes sense or not, right? So it will keep changing. So this is called as insertion and deletion. So insertion will lead to insertion mutation and deletion will lead to frame shift mutation. This way, now if I add G here, right, Ram has a big red cap. Now it makes sense, but still at least this codons will be correct. That means Ram has red cap will code correctly. BIG will be coded separately. So if insertion happens, 3 insertions should happen. It means 3 properly, one triplet should be there. Deletion happens also properly, 3 should be deleted. Then the issue is kind of in the standby. This is all about your insertion and deletion. So understand, students, tRNA, tRNA is a clover leaf model. All of you knows, I am sure all of you know about this. Now if I am trying to draw, right, this is how your tRNA, oh no, 4 hands. Try to draw it a little more smaller. Right, this is how your tRNA will look like. It won't look like a cross, okay? It will look more to one side. Wait, I'll try to draw it once again. Yeah, try to one hand will be bigger, students. One hand will be bigger. But is this diagram visible to all of you? Okay, is this visible to all of you? Yes. Okay, it will not look like this, but yeah, we can still. Okay, this is how it will look, and on the top it will be like this, and here there will be something else. Okay, this is how your tRNA will look, right? So this region, right, this lower portion which will be going to read your mRNA will be called as anticodon loop. Called as anticodon loop. Is this visible? Yes, it is visible, right? Anticodon loop. This will be called the T loop. Okay, here this will be called the D loop. And here you will have your amino acid acceptor. Okay, this will be your amino acid acceptor site. Okay, here is where the amino acids will be accepted. Clear? So here like this we can draw. Yes, this is your amino. This is how your tRNA will look like. Okay, tRNA will look like this. You can even label the 5' and 3' end. Okay, yes, fine. Now what is going to happen if, so imagine you have a codon here. Okay, yeah, imagine A, G, U, it's like this. It is there. This will be what? U means T, no? A. It will code for A. So it will form anticodons, right? Anticodons. Okay, I'll erase this. Oh no. I don't think it will come back like that. Okay. tRNA went. Yes, this is what I meant, students. So here A will be actually forming for what? It will be for U on the mRNA. Okay, on the mRNA. So here what will happen? Phenylalanine will be binding. Okay, so that is the amino acid acceptor site. Yes, so we did a great class today. We understood a lot of important topics, and I have also written the notes for you people to copy down. Okay, yes. So yes, students, we have understood all the experiments, and we have also understood transcription properly, and now we have translation, DNA fingerprinting, HGP project, lac operon. Then we will be done with this chapter. Okay, I'm taking the chapter slowly because you have still time, and most of you would have already done this chapter 2-3 times because it's a very high weightage topic, so there is very less chances that a student wouldn't have gone through this chapter. So this will be like a very slow revision for you people. So yes, thank you so much for joining, and let me know in the comments if you found the class helpful, and if you want the notes, please do join on biology. Okay, 1 second. Okay, this is the Telegram link if you people want to join. 1 second, I'll tell you the Telegram link, students. Give me a minute. Yes. Okay, this is the Telegram link if you want to join. Okay. _NEET. Okay. Or else easier way to search. Come to Telegram and search Biology by Gopika. Okay. And you will be in my channel. Okay, all the notes will be shared there. Clear? If you want any particular notes, and students, definitely join Vedantu NEET English. If you want any notes, let us know in the comments. Okay, thank you so much for joining. See you all. Bye. Take care.